My name is Dr. Chenji Lin. I am a professor in the Department of Biomedical Engineering at IUPUI. The main focus of my research is on designing multifunctional and highly responsive polymer hydrogels for tissue regeneration and cancer bioengineering applications. Specifically, we have three areas of work. One is on pancreatic tissue engineering. The second area is on designing materials to deliver drugs for treating cancer metastasis. And finally, we are using our materials as a bioink for advanced biofabrication. In the area of pancreatic tissue engineering, we are working with differentiating pluripotent stem cells into pancreatic organoids. And in this area, we are interested in seeing how material properties, how local matrix properties can direct and influence pancreatic organoid development. And because of our ability to control material properties, we can actually mimic how a cancer cell microenvironment evolves over time. And from there, we study how cancer cells respond in terms of their proliferation, in terms of their drug responsiveness. And using our materials, we are creating injectable polymers where we can load and release therapeutically relevant agents such as proteins, peptides, or synthetic drugs into the site of disease. Particularly, we are interested in treating metastasis because it is highly lethal for any cancer that metastasizes. And so finally, our third area of work is on biofabrication, where we use our materials as a bioink to bioprint. Bioprinting is a process where you print three-dimensional structures in the presence of cells. And because of our ability to synthesize materials with highly diverse, highly responsive, and highly biocompatible properties, we are able to use these materials as bioink to fabricate complicated geometry of materials or scaffolds through bioprinting, or we can simply use microfluidic devices to fabricate microgels as building blocks to create higher order structural scaffolds. So in terms of teaching, I am teaching tissue engineering, and this is a senior and graduate level class where we discuss how tissue develop and what are the properties of the tissues, as well as how we can use engineering tools and materials to help tissue regeneration. The second class is cancer engineering, where we discuss how basic principles of cancer development and what are the engineering tools and methods that we can use to study, to diagnose, and to treat cancers. And finally, the third class is an advanced 600 level class, engineering principles for biomolecular interactions. And this is basically studying the science and engineering behind receptor ligand binding and how that binding eventually leads to changes in intracellular signaling and how that leads to microscopic changes in cell behaviors. And I hope you enjoy IUPUI, and I hope you consider IUPUI as your college destination, and I hope to see you soon. Thank you. Bye-bye.
Hi everybody, my name is Matt Arkenberg. I'm a third-year PhD student here in the Lin Research Group in the Biomedical Engineering Department at IUPUI. Our research group focuses on using multifunctional polymeric biomaterials for tissue engineering applications and for developing three-dimensional cell culture platforms. My research specifically focuses on using chemically defined hydrogels to investigate induced pluripotent stem cell differentiation and culture in 3 dimensions. We have a breadth of research projects currently going on in the lab, ranging from studying pancreatic cancer in 3 dimensions and developing biofabrication processes for generating microgels for various applications. These projects are highly interdisciplinary and require using tools and knowledge from polymer chemistry and engineering to molecular and cell biology.
Hi, my name is Han Nguyen and I'm a second-year PhD student. So my current project is focusing on studying osteocyte and pancreatic cancer interaction. So what I'm doing here is actually using this live imaging microscope. I want to monitor the migration of pancreatic cells. And as you can see here, this microscope can take image of the pancreatic every hour. and I can kind of see the migration of pancreatic cancer cell.
Hi, I'm Fang Yilin, a postdoc in Lin's group. One of the research area our group is working on is using hydrogels to direct cell fates. So what we want to do is modify the material with different functional groups so that our hydrogel can have different properties. After we have our materials done, then like this one, we have the hydrogels on the stage here, and by using this rheometer, we can test hydrogels mechanical properties, like how stiff are these hydrogels and are they viscoelastic. This test here is we control the temperature of the stage, and what we want to see at what temperature the gel starts to stiffen, so like it cured. So like for this one, it cured right about 30 degrees C, and this is just one of the testing we do for our hydrogels. We have other testings going on as well.
Hi, this is Minnie. I'm also the postdoctoral researcher in Dr. Lin's group. My current study is using the 3D bioprinter and fabricate the 3D scaffold or the 3D construct to cell culture system. My research showed that I developed the new bioink system using the this group's chemistry. In this research showed that without the light mediate cross-linking, the 3D construction is not stable in the room temperature, but after using the light cross-linking, the 3D construction shows a little bit more stable and a longer period.
My name is Lim Lab, and I've been working here in this lab for 2 months as a new postdoc associate. So my mission here is to design and fabricate microfluidic chips. So as you can see from here, we use these chips to make either microcapsules or micropatterns with single cell resolution and on microlin scale, so that we can use these solid and hydrogel features for the creation of highly structured, multifunctional tissue scaffolds.
Hi, my name is Wilbur Chang. I'm a second-year PhD student. My current research focuses on mimicking tumor microenvironment using a hydrogel-based system. This is a nanoindentor. This instrument works similar to an atomic force microscope. So it indents the surface of the material and measure the Young's modulus. This nanoindentor allows me to measure, for example, stiffness gradients across the surface of hydrogel.